Hello, and welcome to demola. And today's lesson we are covering epigenetics and polygenic inheritance. Should be quite a quick one. Let's get it out of the way and get out of this topic. Let's go.
Okay, so we're kicking things off with polygenic inheritance. And specifically, I want to define two terms because I cannot emphasize enough your inheritance terms are so key. So your genotype is the first one we're going to talk about. And this is the combination of alleles at a specific gene, whereas a phenotype is the observable characteristics of an organism. And what you need to know about phenotypes is it's all going to be to do with gene expression. And we're going to talk about that a bit more when we get into epigenetics. But it's about the expression of your genes. Now, some characteristics are controlled by continuous variation, some are discontinuous. And we just need to be able to tell the differences between the two and know which characteristics or traits apply to each one. So when talking continuous variation, we're thinking height, weight, skin color. Skin color is a bit borderline, but the kind of thing they're going to say is about, yes, there's obviously a big genetic component, but then also things like tanning is going to change the skin color. Whereas discontinuous is more like your blood type, gender, tongue rolling, which is one they love, actually, ability to tongue roll. But these things are kind of just like, they kind of stay as they are, if you get what I mean. So, continuous variation controlled by multiple genes. That's where we get our polygenic inheritance, multiple genes. Strong environmental influence, range of varies between extremes, follows a normal distribution, and you represent it with a histogram. If we talk discontinuous, controlled by one or a few genes, little to no environmental influence, distinct categories, no distribution pattern, and a bar chart. So, just to kind of add a bit of nuance to this, say, for example, height and weight. Yes, your height and weight is in some way determined by your parents. As in, if my parents are tall, I'll probably be tall. But also, if I don't eat those greens, yeah, I'm not going to get tall. If I don't have my calcium, I'm not going to get tall. So it's kind of things like that with continuous variation. Whereas discontinuous, my gender is solely completely determined on like the combination of your chromosomes that you get, X, Y, X, X. That's not going to change, essentially, from a genetic standpoint. So, continuous variation, you can see we'll use a histogram. So histogram, you'll see that the bars are kind of joined together. And it does follow a bell curve kind of shape. I'm not going to be able to draw it well, but this is kind of like your bell curve. And then your discontinuous variation, there's not really a pattern to it. It's a bar chart, so you can see the bars are separated. And yeah, that's about that. So just be aware differences, continuous and discontinuous variation. Okay, so now we're going to introduce a new concept, epigenetics. And this is how gene expression changes due to environmental factors. And I'll just throw a couple out there. Smoking, stress, exercise, diet. These are just some environmental factors that can affect your gene expression. Now, very importantly with epigenetics, we need to be certain that there is not a change in the base sequence of DNA. So we're not having a mutation. A mutation is a change in the base sequence of DNA. That is not happening with an epigenetic modification. In your eukaryotic cells, your DNA is wrapped around histone proteins. We should have covered that in the cell structure video at the beginning of this topic. But we do not have, so we have it wrapped around histone proteins. And the histone proteins are modified through or how tightly they're wrapped around the DNA, I should say, is modified by the addition of methyl groups and acetyl groups, which I have drawn underneath. So whenever you're answering an epigenetics question, really, I want you to follow these kind of steps or principles that should help you to get the answer every single time. So in DNA methylation, you're adding a methyl group to cytosine, specifically cytosine. I'm going to underline it because it could come up, not necessarily the most important thing, but it's nice to know. So you're adding methyl groups to cytosine bases. So your DNA is going to wrap more tightly to the histone protein. Yeah. Remember what we were talking about when we were covering like operon. We have a promoter region on your gene. Yeah, and this is kind of why your transcription factors and your RNA polymerase are going to join to activate that gene. So if DNA is wrapped more tightly to the histone, the promoter area is less accessible to RNA polymerase and to the transcription factors. And if that's the case, your gene is less likely to be activated or less likely to be expressed or less likely to produce protein. They can use all those terms interchangeably. That is what happens when we methylate. When we acetylate, it's kind of the opposite effect. So you add an acetyl group to lysine. Yeah? DNA is going to wrap less tightly around the histone. Promoter area is more accessible to RNA polymerase and transcription factors, so your gene is activated, more likely to be expressed, more likely to produce your specific protein. So just know that's the order you want to go for an exam. You should be able to get 4 marks every single time using that. And something that you need to be aware of as well is that when cells divide by mitosis, they can actually pass on their epigenetic modifications. So, for example, the methylation patterns are copied. Let's say, for example, I've drawn some DNA. That's your parent cell. These are your daughter cells, right? I'll put the P and the D on the same side. Cool. What we're saying is, say if in red, I say this gene gets methylated. In both of the daughter cells, it's also going to be methylated, hence less likely to be expressed. We don't really talk about it in terms of acetylation, so just kind of remember that. But you can pass on those modifications, so that's going to, in a way, affect how genes are expressed in your offspring as well. I also would like to add, there's a very, very important gene for breast cancer called BRCA. So BRCA1/2. Its expression is controlled by epigenetic factors. So things such as your diet, smoking, they can affect how this gene is expressed, which influences how likely a woman is to develop breast cancer over their life. So just remember, that's kind of a real life application of what we're learning here. Like I said, it's a nice quick vibe today, and we've got 3 questions for you to try. So the first one is talking about epigenetic changes. The next one is talking about how adult height might be greater than the biological parents. And then the last one is kind of a data question. And you're looking at the data and commenting on the effects of genetic and environmental factors on the body mass of twins. So I'll zoom out over all of them. Hopefully you can see all of those. I'll give you some time, pause the video, and we'll get into the answer. Okay, answer time. So first one was just talking about epigenetics, and honestly, like I said, go through the same kind of structure. So say we chose methylation, DNA is methylated, meaning that your histones wrap more tightly to the proteins. That's kind of, your histones wrap more tightly to the DNA. That's the first thing it's saying. Then we were talking about the promoter region becoming more accessible or less accessible in this case to RNA polymerase and to transcription factors. That's hitting your second marking point here. Yeah. So we can also add in a part about activation or deactivating genes. And then if we deactivate a gene, it's going to affect the protein produced. So that's going to affect the enzyme production metabolism. I'm not sure if you got the context in the question, but just kind of imagine, always apply context when you're answering questions. Next one was talking about how an individual can be taller than their parents. And we should know it's one of the more common examples that height is polygenic inheritance. So it's affected by environment and genes. Cool. So yes, there's going to be multiple genes that are going to influence the height, but also environmental factors. Like we said, calcium, get those bones strong. Protein is nice. Healthcare probably can make you taller. Yeah. So just think about stuff like that when you're answering a question like this. Now, this is the big one. If we look at the graph, what are we kind of seeing here? We're seeing in the monozygotic twins, fun fact, such as myself, we're seeing in them that we're hitting a lot more of this like 90% region in terms of the concordance. Yeah. But we aren't seeing anyone that has 100%. So none of the twins have the exact same body mass. Whereas you can see here, if we're looking at the dizygotic twins, there's definitely less of them having us high up. So in terms of the percentages being like correlated quite the same, there's less, right? And you can also see there's more of like a downwards trend here. So those are the kind of things we need to spot on the graph. So when you're going about answering this question, we're definitely going to want to say, well, monozygotic twins, they have the exact same DNA, right? So we've said higher. That was the observation. That was higher. Then afterwards, we're going to say, draw a conclusion, because we're commenting, right? Genetic factors have a greater influence. And that is because we're going to give evidence to our claim. And the evidence is that the explanation is because of the identical alleles, right? Cool. Now, we also pointed that none of them were 100%. So none of them being 100%, what does that mean? Explain that. It means that there are environmental factors that affect the mass. So I hope that's made sense in terms of my explanation about how to answer this question. Okay, and this leads us to the A-star tips. So to be honest, they're a bit wishy-washy today, but things that we kind of want to be doing is identifying continuous and discontinuous variation. So think about the characteristic, which one would it fit into? Polygenic inheritance, just be confident with defining it. I think when you really look at the word, it kind of gives you the hint. So poly means many, and then genic is just talking about genes, so multiple genes, and then that should be fine. And then methylation and acetylation, that kind of 3-4 marker, I'd just say memorize just doing that, and you'll be able to get the marks most of the time. Other than that, I guess knowing the normal distribution is quite useful. So normal distribution, I did stats, so it's kind of got drilled into me. But it does kind of look like that, like a bell curve. Mine isn't the best, but you can see it kind of looks like that. And then when you talk about like mean, median, mode, they're all kind of in the middle if we don't have any skew. Now, stages of protein synthesis is also really important because they can easily go on from a question about methylation and acetylation and link that to DNA replication, or sorry, transcription. So just try and link those together, right? I think, especially in a course like SNAB, they really do like those links between topics. So just be aware that's something that they will do. Anywho's, hope that's been a very quick video. I hope you liked it. If you did, please like the video, drop a comment below, and subscribe to the channel. We are very, very, very close to 800 subscribers, which is absolutely amazing. Thank God. So hopefully we get there soon. Any questions at all, drop them in the comments below. I'll do my best to answer. We've also got our TikTok and our Instagram, both at @demola, and drop them a follow. That'll be really, really appreciated. We'll catch you in the next one. Bye for now, and thanks for listening.